Good afternoon, everyone. I'm just very glad to be here. Thank you for the invitation. I'm Ana Filipa. This presentation is going to be shared with José Montes, and I will be talking about the potential of eDNA metabarcoding as a tool for early detection of non-indigenous freshwater species. Okay, I'm from CVU in Bio and ISA. So, a summary of my presentation, just for you to know what to expect. I will talk about the background, about freshwater biodiversity and the non-indigenous invasive species problem. Then about the approach, what is the eDNA metabarcoding and which are the steps that we must follow. And then I will talk about the validation case, and Jose will develop this part. It's the more technical part and online part of it. So then I will make the conclusions and talk about the challenges. So about freshwater biodiversity, what is need to know? It's freshwater biodiversity. It's the one that is declining more according to this index, the Living Planet Index. So, and one of the major threats, is habitat loss, but then you have the problem of the non-native species, invasive ones. Okay, so this is the thing that you need to know. So what is this problem of non-indigenous invasive species? Because actually, since humanity, this problem of transporting species is an old one, but it was in 1958, that Charles Elton just made the early warning about this problem of invasive species, and then now it's growing exponentially. So there is this problem of biological invasions. So non-native species, this is about freshwater, this map is about non-native freshwater fish. You see that in red, the most of the species are nowadays they are not native from the region. They have crossed oceans and lands to be in other parts of the world. So, for example, in Portugal, I'm from Portugal, that's why I'm talking about this. 70% of freshwater fish are endangered. Okay, and then you have this relation that the biggest the number of exotic species, the decline is bigger. So about the environmental DNA, what is it? This thing that everyone talks about, it's the DNA traces in water, feces, bulk, sediment samples, and that we can, with this environmental DNA, we can detect species and do other studies, other ecological studies and genetics also. So this is the classical approach, is the barcoding. What we do is that we compare the traces in DNA from the sample to reference libraries of DNA. We choose a marker that is the most appropriate for the taxa, for the species that we want to discover, and then we compare with the reference database. So DNA metabarcoding has all these steps. You can collect, for example, in this example, you can collect water, then you filter this water to have all the DNA concentrated, the eDNA concentrated, and then we extract, amplify, and then put in a machine, the sequencing machine, to have all these the identification of all a bunch of species, not only one, but a bunch of species, and then from these species we can. It's our aim in this work to identify the ones that are non-native. Okay, this is the process. So in practice, this is a multidisciplinary work actually, because we have challenges in the field. Where should we collect this water, for example? In this case, is water we are looking for. Then we have many laboratory steps, and then we have the office. I put here below this. You have this figure. It's only for you to know the different paths, the different methods that you can use to until you go to the sequencing. So there are lots. This is a technique that it is developing, and then everyone working in this is trying new approaches and new ways of doing this, just to obtain the best results. Then after all these challenges, then we have the bioinformatics, and here it's where LifeWatch, and in this work we are we are trying to develop. Okay. So I give you one example just for you to understand some details. This I will talk about in the conclusions. But here we are just it's a study about barcoding. We just discovered a species, invasive species in Portugal. It is this green thing. And when we were looking for this marker in DNA, we cannot find anything similar. Sorry. When we look at this marker, this part of the DNA, we can see that Charente River, it's the populations are very similar to these ones in Portugal. But when we look at this marker, the other one, we can see that. Sorry, I have so many windows in my computer. We can see here that because people did not sample here in the Charente River, we don't have any comparison to know what this is. So the take-home message is it depends. The result it depends a lot of the marker that you use. So in this study we were able to say that this new species, this non-invasive, non-native species, is from the Charente River in France. So the aim of this validation case, the main aim, is to detect new niche species with eDNA metabarcoding, as I told you. So we have several steps that Jose is going to describe, but the detailed aims are to run the pipeline of taxonomic assignment of a metabarcoding run, produce a list of NIS species, from a sample, and of the remnant species also, the native and the new niche for that sample. Build the mapping and then see which things overlap all the results and see which occurrences are outside of the current known distributions. So I will give the floor to Jose now.
Thank you, Filipa. And I'm talking to you on behalf of the technological part of the Lightwood Jared ICT core, and in order to give you some details of this metabarcoding workflow. The aim of this workflow, as you know, is to detect likely non-indigenous invasive species with metabarcoding, and very briefly, as you can see in the presentation, in the first step of the workflow, the eDNA metabarcoding sequence are computed in step 1, and then they are checked if they are listed in the non-indigenous invasive species in a given country, because this is dependent on the location, and at the same time, if they are also available in GBIF, and this is step 2, then the occurrences of the sequences which are not classified as NIS are extracted from the GBIF database in step 3 and are intersected with the location of the species that are not belonging to the NIS checklist in step 4. Okay, if any occurrences are found, the given sequences that are identified are tagged accordingly. For example, based on the occurrence distribution, we may say that a sequence is likely to be a new non-indigenous invasive species in step 5. So as you can see, this is somewhat the implementation of the workflow. Okay, can you pass to the next slide? Okay. Here is what I was describing in this diagram. Shows you in a more precise and concise view the implementation of the metabarcoding and how it shows in the Tesseract graphical user interface. You can see that there is a direct correspondence between logical steps and the representation in the graphical user interface. Okay. This metabarcoding workflow, in fact, consists in 5 services which are running in sequence. This means that there is no kind of parallelism in this workflow, and neither we are invoking an external resources, or there is not interaction with the user or whatever. You should know that in the very first step, namely the metabarcoding runner, that we are going with, which we saw before, comprise 8 minor stages, which are manually carried out by scientists. This means that this is the implementation of the workflow, but in this case, the implementation of the workflow needs an input from a scientist, but it hasn't any interaction during its execution. Okay. As a result, the outcome is directly imported into the outcome of the previous step made by the scientists are directly imported in the workflow. So for doing these things, we will wrappers to abstract the underlying implementation of each one of the services, and we provide the scientific teams an abstract interface for each one of these services in order to hide completely the complexity of the underlying implementation. The scripts or the code of each one of the services are mainly developed in R, the R programming language, and in some case, these R scripts have conflicts. This is because of this, we are using containerization. We are using containers for the code in order to encapsulate all the scope, of the execution of each one of the steps in this workflow. This is an interesting thing because the many steps of a given workflow, and this is the case in this one, can be developed separately and with very different orientation and objectives. And you can reuse this code and also the component that encapsulate the code for its general reuse in order to take advantage of the previously built script or algorithm, and also we you offer it to the community to build new workflows. Can you pass to the next slide, Filipa? Thank you. Giving some more detail about the service isolation and why we're using containers, what we do is create with the algorithms and the script or the code, whatever you want to call it. We create a Docker file and put it in the Docker container, which is somewhat like a black box in which you are able to encapsulate all the dependencies of the context for the execution of this piece of code. Okay, this means that even if you could have any kind of conflict for the several services that you are using, for example, because you are using different versions of R libraries, or you are using different versions of whatever other tool that you are using, like GITAL or whatever. These conflicts are completely avoided by the containerization of the algorithms. Okay, so with this architecture in mind, we can potentially encapsulate any services developed within any platform and executed these services which are incoming from different environments and high heterogeneity, but also we have the ability to deploy these services in different environments. This is also interesting from this point of view, not only to compose the services and run them in a single environment, but being able to encapsulate the services, we can execute the service, for example, in a local machine. We can hire resources to the cloud and execute the component, the Docker component remotely or whatever. And this is quite flexible. So as I had mentioned, we have developed wrapper for each one of the services of the metabarcoding workflow, and this component communicate with the Docker engine to manage all the life cycle of the containers and the workflow, and this leveling of encapsulation don't require that all the components and all the workflows are fulfills with any dependency. So it gives you a great degree of freedom at the task of integrating heterogeneous things. So I think that this is the idea from the technical point of view of what we did. And thank you. And I give back the floor to Philippa.
Very nice to see the advancements of this part of this work. I'm pretty happy with this. So I will just make a wrap up about some conclusions that I thought, just for sharing with you. So what up to now, it seems that this method, eDNA metabarcoding, allow us to sample the whole fish communities and to identify new niche species, the old and the one, and the news, actually, the both. So this technical, this tool that we are producing here with LiveWatch will help to this difficult task of bioinformatic treatments of samples. Well, with this tool, for sure, and it will be possible to scale up the use of eDNA metabarcoding for monitoring large regions and respond to EU directives. At least that is what I think that might happen for the next years with the broad use of this tool. So what I see from the last works made on this subject is that combining multiple methods it's one of the ways that this investigation is leading. Is like eDNA metabarcoding is not going to replace samples with electrofishing or samples with nets. The thing is, these approaches are quite complementary. That's what it seems up to now. Challenges that we must face. We have lots of challenges to think about. One of the things is just to have this automatic link to online eDNA reference libraries as BOLD and GenBank, and also GBIF, of course. Should be flexible enough to incorporate a user build-up DNA reference library. I don't know if this is still on the table. And the thing is, as Jose mentioned, is because we have this, the user can have a role on this, the first step of the pipeline of the workflow. Then we can have these several pipeline options for filtering this sequencing data. It could be nice to think in the future of a kind of certainty index to say, okay, we are certain that this is a new NIS species, or we have many doubts of it and we need more information as photos or another kind of observations, direct observations of the species. So it's this that it's again combining multiple methods might be the best solution for advancing with this research. So I'm finished. I'm just acknowledging all the projects that we have at CBU involved in this for the last years, and also LabWatch for having this very intense collaboration and very fruitful collaboration. Thank you.
Thank you, Filipa. As we always say, LabWatch is a humble servant of the communities out there. This is our job, to facilitate their work. Now, our next speaker comes, from a project which is called Life Mika, and it's about it is going to be demonstrated by Miriam Boonstra, and I guess that Miriam is with us already, and she prepared.
Yes, can you see me?
Perfect. Yes.
Yeah, so I'm from a different live project. The project I'm in is Live Mika, which is an international cooperation between 3 countries, the Netherlands, Belgium, and Germany. So Mika stands for Management of Invasive Koipu and Muskrat. It involves various organizations and institutes of these countries. The aims of this project is developing new methods for better management of these invasive species, also reducing unwanted side catches. There's also a part on the effect on the ecosystem, data gathering and sharing and of these 3 countries on the presence of these species, and also importantly monitoring of public opinion and outreach on the management of these invasive species, because animal rights groups can sometimes be opposed to this. And what we are, the eDNA is part of the improved management part of this project. It's mainly a cooperation between the Dutch Genomic Service and Support Provider of the University of Amsterdam and the several Dutch water boards. There's a reason that this part of the project is initially being tested in the Netherlands, and I will tell you why. Let's see, how do I get to the next slide? Okay, wait. Yeah, so you can all see the next slide now. Can you still hear me? Yeah?
Yes.
Okay.
Yes.
Okay, great. So a little bit about the Netherlands. There's about 300,000 kilometers of waterways in the Netherlands, and a large part of the Netherlands is sensitive to flooding. Therefore, muskrat and koipu trapping is done by professional trappers. These trappers, they work for the Dutch water boards, who manage most of these the waterways in the Netherlands. The cost of this management is approximately 33 million euros per year, so that's huge. And 70% of the cost goes down to into tracking and muskrats. So the trapping itself is a small part of the cost. And most of the cost goes actually into tracking them. So tracking down muskrats and koipu is a highly specialized skill. It's not easy, and therefore we are using the Netherlands uses professional trappers. So the main reason why we want to try to use eDNA is to reduce the cost and time it takes to basically track down these species and allow the trappers to focus on catching the animals, because there are some signs that there's been some selection on the animals as well, where there are now animals who can recognize and avoid traps. So there might have been some selection of smarter animals. Because the added value of the eDNA method is primarily in areas where the population of muskrats are low. We are starting at least the muskrat part of the project in two provinces of the Netherlands, the northern part of North Holland, which you can see here, and Friesland. These, in particular, Friesland has a relatively low population of muskrats. They've been very effective in removing them. And how are we approaching this? The eDNA part. In some initial pilot tests, we learned that it's feasible to divide the Dutch waterways into routes of about 3 to 5 kilometers. And of these waterways, about 500 ml of water samples are collected by an automated GPS-based water sampling device. And after one of these routes is, our sample of these routes are sampled, they are analyzed by qPCR. And then if a route is positive, we are now having the approach, which is subject to change, is that the route is then sampled with 1 kilometer subroutes to identify the linear home range of the muskrat. And then to further narrow it down, we do point samples of 100 meters to localize burrows. And the next step then is to place the traps, catch the muskrats, and afterwards checking if all the muskrats are gone by eDNA sampling. Now, when I say this is subject to change, it's because the situations in the fields are very complex. There are many factors that are have to be taken into account. So while we know that the first part of the method is working, the 5 kilometer subroutes and the eDNA part, how we then approach these follow-ups is right now being tested. And this is done in very close cooperation with the trappers, so that makes it quite interesting. It's not a typical scientific project where you have your method set out and then you do the experiment. This is, we are relying on their field experience, and then we, if is necessary, adapt the approach. And in order to facilitate the sampling, we are now also working on several other tools to make it easier for them, like a remote controlled boat on which the auto sampler can be placed, and also sampling, for instance, from a backpack, because not a large part of the waterways cannot be sampled by boat. And so this is why we are working on that as well. And yeah, what else would I want to tell? That is basically the overview of the project. So right now, testing of the field approach, and I'm keeping it short. So, like I said, this is a cooperation mainly between the water boards and the University of Amsterdam, at least this part of the project. And if you want more information on the other projects within Live Mika, you can go to their website and get some information there. So if there are any questions, hopefully there are some questions.
I can see some comments that are coming up in the box. First, it comes, from Eliana. She congratulates Filipa and Jose for their talk, and the question is, how easy would it be to extend this workflow to other environments, taxonomic groups, as it is now currently implemented? I guess that this question can be replied by Jose first, and then Filipa.
Yes. Okay. From the point of view of, from the technical point of view, extensibility is quite easy, because what we are developing everything from the point of view of reusability. So there is no precisely what we are doing is trying to encapsulate analysis algorithm and each step of the services in a technical wrapper that allows for portability. And also we are making a second dimension, which I don't talk about before, that this is the semantics, the semantic annotation of inputs and outputs of each one of the steps. We are clearly defining what we are expecting in each one of the components. Once we take an algorithm and we put these algorithms into a wrapper and we hide technological complexity in this black box, we are also annotating externally inputs and outputs and requirements for the execution of this component. And we are also annotating semantics of input, not only what type of data we are expecting and CBS a file or something like that, but also the semantics of each one of the attributes that we are expecting. It's this kind of concept that is well defined in this kind of in this ontology. For example, in this case, the live ontology, in which you are defining the semantics of the inputs and the outputs. So the class to which belong each one of the concepts that we are using in this step. So everything is designed to reusability. So once you have a component developed, you can share this component in quite different workflows, which are designed with quite different objectives. So this is not only about the reusability of a component, but it's also about the reusability of a full workflow, because at the end, to us, a full workflow or a full branch of a workflow can be encapsulated in a component that is also reusable. So you can see that everything that you can see in this workflow editor or in a workflow can be reused and encapsulated in another component and reused in several contexts with a well-defined context and metadata and semantics.
That's the most important aspect of all this thing. I will come back, after the reply of Filipa. Filipa, please go ahead.
Yeah, thank you, Jose. The semantics is, I'm getting aware that semantics is really important here in this tool. The thing about the scientific part, yes, I believe that all these steps are common in different taxa for different regions. So the challenges that we have, it's for some taxa, the reference databases are really poor. Actually, this kind of developments of eDNA metabarcoding are mostly in the northern hemisphere. So more than half of the world is completely without studies about eDNA metabarcoding. So there are regions that they are quite unknown for reference databases of DNA. Of course, this is you have the problem of taxa, you have the problem of regions, and also if you change species and your target, you must use the correct marker, genetic marker for targeting your approach. So it's a challenge for the 21st century, for sure.
Thank you, Filipa. I'll pick up, one of your points, and I will start, talking about the dark biodiversity material, because this method actually can be applied and can have tremendous results and tremendous and huge rivers of information that can describe, this dark biodiversity material. And I will pick up, the point from Jose, where the tesseract composability layer. I tend, to look at that like it is, not just a VRE, but just a platform that enables VREs, to come up and be combined. And the beauty of that is that any service that is being deployed there, it can be combined with any other service which has done, the same thing and has been landed on the system. And that for Ileana means that we may have, combined approaches. For instance, yesterday we saw, ARMS, which engages two approaches, one on the metabarcoding and another one, the conventional community analysis. And, at the end, the patterns that can be derived and the processes that can be inferred from both approaches, they can be compared to each other. And surprise, surprise, as Matthias said, the first results, the first attempts, tend to show that the patterns are not similar. They are rather, complementary. And that's a fantastic opportunity, for the biologists to go hand in hand with the computer engineers in order to develop, the material of the next generation biologists or scientists on biodiversity and ecosystem functioning. One other aspect that probably needs to be brought for more discussion for the both of you and also for Miriam is to what extent the environmental information can come to this play, and what kind of questions like those that Mike was referring to in the morning session, the what if and so what questions um, can be somehow answered through this type of workflows. If we don't analyze only biological data, which are coming from sequencing, but also combine this data with environmental data.
Of course.
What I mean environmental data, mostly abiotic data.
Yeah, of course. That is the most interesting thing for me, that I'm an ecologist. It's just to see the changes across time, and changes in a very disturbed ecosystems. And so if we are able to develop this eDNA approach, we are able to monitor timely the ecosystems and predict a little bit the destruction of ecosystems. Of course, that is quite interesting. I hope we can achieve that. Of course, that is the very interesting question. Thank you.
Yes, thanks, Felipa. There was, some intention behind, but I will come back to this in a minute. Jose, for you, the question is this feasible through the workflow? Is this feasible, to take into account, to engage not only DNA sequence data, but also environmental data? Is it possible?
Sure. You can integrate whatever kind of open data of reference, like GenBan, as said Filipa before, or like weather and climate information, and also information from sensors or whatever you need. And also the interesting thing about this is that once you have integrated one data sources, what you have is a component that can be reusable in any workflow that in which you could need to use this. So this is a very efficient approach to integrate this new kind of data into the research tasks of the scientifics, because once you have a component to access or to link GenBan to access for some sequences or protein function or whatever, depending on the database that you are looking up, you have this component available for anyone who would interested in using it later. So all these kind of things are feasible. We already had done some of these tasks in some other areas in system biology or something like that. So I know really that are feasible. And once you have integrated in a component, you can have it available for anyone who is interested in using it.
Thank you, Jose. Fantastic attempt to take examples from the system biology, because so far they are the ones who go first ahead, and we need, to follow, the trends that they become, very useful, and the examples that they become very useful. Miriam, would you like to add something on this line. In brief, the question was, what if in this workflow we can include information and analyze information from the environment, not only from sequence data, but also from the environment, environmental variables like the temperature or the humidity, this kind of things.
Well, I mean, it's not an integrated part of our workflow, but we have to take into account conditions in the field. So, for instance, seasonal changes are very important. I mean, we are running into practical things like water plants and stuff like that. Yeah, and the weather plays a role, and also the size of the waterways, the flow, the currents, and things like that. So we try to get some data on it, but it's not an integral part. In that sense, it's more practical. What you can do with this method itself, which is maybe for the future interesting, is that you are collecting 500 mL of water over a 5-kilometer waterway area. You could gain information about pollution, other species, other invasive species, in endangered species, stuff like that. I mean, that is what you could do with the sample itself, but really kind of in the workflow itself, like environmental is not an integrated part. It's more a practical approach to it. I mean, we want it to work, and we try to approach it in as practical way as possible, because we are basically working with two very different organizations, and it's more on the practical part than the scientific. That's where the challenges are.
Thank you, Miriam. I will go back, just for a minute to the reply that was provided by Philippa, because she touched upon a um, a problem that we very often, have, which is how much of the outcomes of our research here is useful for the stakeholders. For instance, if we come up, with some kind of prognosis, uh, how many and how many invasive taxa that have been identified, through the workflow, the DNA beta barcoding can be uh, can be occurring in the near future in which places, then we can give, valuable information to stakeholders like environmental managers, or even those that are making policies in order to be in order for the society to be ready to tackle these problems, these coming problems, even because now we don't know what's going on. But if we run several scenarios, and the workflow can enable us to do that, then we can give precise prognosis, and then we can solve some kind of needs of this side of the stakeholders. And I think that's very important, because now in Horizons Europe, the whole thing is being changing. It's not science anymore, for making science and making breakthroughs and advancing actually what we currently know, but it's about how to make these advances to serve the various needs of the communities, and of course stakeholders are one piece of this chain, and in fact the EU is going to launch a lot of instruments, like missions, like the European Green Deal calls, etc., etc. So that's very important. I have here in the box one question from Christina Dimuri to Miriam, and the question is, if you, Miriam, have evaluated the cost of tracking muscat and copy using eDNA or qPCR compared to traditional surveys.
Yes, we are now working on a business case, and yeah, if we can indeed implement this method successfully, it will be much cheaper than the traditional method of detecting muskrats. We are still working out the exact details of the cost. I cannot give you numbers, but indeed, if it works, if we can reduce the time that the trappers spend on having to search for muskrat, then yeah, it will be a significant reduction in the cost for the water bars. So, but the prognosis on that will come somewhere during the Live Mika project.
Thank you, Miriam. The next question comes from the head of our virtual Laboratories and Innovation Center, Peter van Tinderen, and it's for Philippa and Miriam, I guess. Peter says DNA is fairly stable. By that also means that the signal can come from organisms that are dead or long ago dead. And if you can make a distinction between the old and the dead and the new and the living DNA. Philippa, first you and then Miriam. Thanks, Peter. Amazing question.
Yeah. The thing is, we can't. I believe we can't. The thing is, DNA degrades a lot when it is in the water. Then dead animals, well, the eDNA from dead animals don't last for long. But this is it's very uncertain. But because if it is in a climate like in Portugal, it's going to degrade faster. But if we are in the north, as in Netherlands, it's going to be slower. This is not really known still. So, but I don't know. I don't know if Miriam has any idea, but I think we don't know in practice. The thing is, mostly dead animals are eaten by others. That's the thing. They don't
Yeah,
last long
Miriam?
in the environment.
Well, the dead animals in this case are not the problem, because if they're dead, they're being removed by the trappers. But we are taking this into account with our method, that for when we do the checks after muskrats have been caught and killed, that we wait for the final for the checking. We wait a few weeks, and even we will have to by testing the methods see what's the best time period to wait between having caught the animals and checking if they're still present. And then we will learn indeed, depending also on, like Anna Filippa said, the weather, how long we have to wait. I mean, that's why we are testing this part of the protocol, and I said it's somewhat subject to change, because we are now learning details such as this, like how long do we have to wait before checking if the muskrats are really gone, to make sure that we are detecting only the live ones and not traces of the ones that we just removed.
But at the same time, this same situation may turn to our advantage, depending on which question we are trying to reply to. For instance, some taxa, some species, some individuals may have secretions that include any type of DNA or RNA. We catch up this in a sample, then we amplify, then we sequence, and then we identify. But still, the animal or whatever else, probably the microbe, is not any longer there. It's been transported by the wind or energetically, and it goes somewhere else, some other place. But still, the advantage of having this kind of information should be that, for example, we have an early warning for a taxon which hasn't been identified before in that area, and therefore now with the new methodology, we could do that, and we could alarm, some of the authorities, some of the stakeholders. A last question, I believe, because we need, to allow people 5 minutes to refresh themselves for the next long session. This is from Kemal Jan Bijel. I guess that I can pronounce his name very well. from enthusiastic challenge to build a firm path towards the future. And he congratulates you guys. And actually, it was a comment and not a question. Well, then, if you allow me, Felipa, what would be, the next step for you?
I think one of the next steps is to scale up, the samples of environmental eDNA. Then we will have, more samples. And to cross with environmental data, as you mentioned. I think with this kind of tools, as the one that LifeWatch is developing, this one, I think we can start scaling up and grow, making the reference collections grow and make this connection with the environment.
Absolutely. And
Yeah.
Miriam, one last word for you.
Yeah, what can I say? I mean, our project is it's a bit different. It's really something that the water boards want to succeed. So it's a very practical approach to a problem that's costing a lot of money. And yeah, it's a very nice project to work on because we're working not just with other academics, but also really people from the field who have their own experience and way of working and knowledge, and we have to integrate the scientific part with the knowledge of the trappers to get it to work, which is is both a challenge and very interesting as well. Yeah.
Thank you, Jose. One final sentence from you.
Okay, I think that this shows that the approach is quite feasible and had many ways to grow up in many dimensions. One of them is censoring, integration of environment data, even proximity and remote sensing. And the other one is omics information and reference open data that are available and can be looked up to extend and boost the analytics we are using in all this data. And then from a technical point of view, all of vaccines are feasible and has demonstrated in some other areas that can be done. So yes, looks like very nice.
Thank you, Jose.